Welcome everybody to, I think, our 12th lecture here in Chemistry 51C. I can't get coffee during this coronavirus shutdown where everybody is social distancing. And I found myself buying an energy drink to bring to work. I don't even know if I can make this possible for you to see. But the energy drink contains something called taurine. And I got interested in that. It's hard not to be interested in organic chemistry, right? So I wanted to find out more about what is this taurine that I'm finding in my energy drink here. When I looked online, I found out that energy drinks can contain over a gram and up to 2 grams of this very simple organic molecule called taurine. It's only got 2 carbon atoms in it. It's got an interesting carbon-sulfur bond. It's a sulfonic acid. You learned how to make arene sulfonic acids using electrophilic aromatic substitution. But this particular molecule has a sulfonic acid residue that is attached to an aliphatic carbon chain, a CH2. And of course, it exists as the Switterion in solution because the molecule taurine contains a strong acid, a sulfonic acid, and a reasonably strong, or a modest base, a good base, I'll call it, an amino group. And so you can't isolate sulfonic acid without having the amine, being on the nitrogen as an ammonium ion. So this is taurine. What's this doing here in my energy drink that I'm drinking? I'm not quite sure why it's there. Why am I drinking taurine? Taurine resembles neurotransmitters that you find in the human brain. Let's go ahead and talk about some of those neurotransmitters, and in particular the fact that they're derived from metabolic amino acids. I hope you all know that there are 20 amino acids that are incorporated into the proteins of all organisms on the planet Earth. One of those is glycine, and in humans, glycine is directly used as a neurotransmitter in the brain. And so essential for neural function. Glutamate is transformed as actually glutamate is a neurotransmitter in the human brain. And that glutamate gets metabolized. I should have noted that on here. And the glutamate, this is basically the salt, the anionic salt of glutamic acid, gets converted by an enzyme into gamma aminobutyric acid. There's a human enzyme that decarboxylates. There's not a simple chemical transformation. We'll teach you for that, but there's an enzyme that accomplishes that. And it makes gamma aminobutyric acid, which is also a neurotransmitter. So your brain has all of these neurotransmitters for Switterions, where there's a plus and a plus and a minus. And maybe it seems reasonable that when you look at the chemical structure of taurine, it resembles these other Switterionic neurotransmitters so much, you can see why people would think that, hey, it's probably got some sort of neuronal function. And just like GABA, taurine is derived from the amino acid cysteine through a multi-step enzymatic transformation that involves decarboxylation, followed by oxidation of the sulfur atom to a sulfonate or sulfonic acid residue, which ends up being deprotonated to the sulfonate. So is taurine a neurotransmitter? Well, when I looked online to figure out whether I'm drinking over a gram or 2 grams of this compound taurine, is that a neurotransmitter? Is that why they're putting that in my energy drink? So I think it's obvious that the word taurine is derived from the Latin taurus, which means bull or ox. And taurine was discovered in ox bile, in oxen, like in 1827. It's hard to believe that they understood enough about chemistry back then to know that they were isolating a unique substance. Bile comes out of the liver in mammals. You've got a liver that's part of your digestive system. It's kind of your molecular trash disposal unit. The bile goes out of the liver into the duodenum. That's part of the small intestine, and eventually comes out your bottom. And that's going to be true for oxen, and it's going to be true for humans as well. And when I looked, I couldn't find any evidence whatsoever that there is a taurine receptor in the human brain there's a glycine receptor, there's a glutamate receptor, there's a gamma aminobutyric acid receptor, a GABA receptor. There's no taurine receptor in the human brain. I'm kind of beginning to think that what I'm drinking is 1 to 2 grams of what's really metabolic waste, and it's going to be quickly removed from my system, just as it is removed from an oxen or a bull's digestive tract. I did see one interesting, I'm not against drinking metabolic waste as long as it doesn't harm me. And I did see some interesting study that suggested that these amino groups react with sugars like glucose to inhibit effects of diabetes where you have poor control over glucose levels. Maybe that's a good benefit, but okay. Let's drink up here. Maybe it's something good for us to drink, is to be drinking taurine in our instead of drinking coffee. We're going to talk about an important mechanistic intermediate that's en route to formation of acetals from aldehydes and ketones. So earlier I gave you a transformation where we generated acetals from aldehydes and ketones, and I told you that you had 2 equivalents of alcohol or 1 equivalent of a diol in order to generate an acetal. But if we look closely, there's an intermediate midway through the mechanism. It's a multi-step mechanism that's an important mechanistic intermediate for us to discuss. So the first addition of an equivalent of alcohol, kind of like the formation of a hydrate, looks something like this, where there's 1 OH group on the carbonyl carbon, and then there's 1 alkoxy group on the carbonyl carbon. And you can see the elements of H and OR that have been added across that carbonyl group. And so at this stage, you still have 1 equivalent of alcohol, and mechanistically, you do an SN1 substitution of the hydroxy group. These reactions work much faster, of course, if you have a strong acid catalyst that's present, like toluene sulfonic acid, for example. That's the one that we discussed. And let me go ahead and tell you the name of this intermediate, because we're going to mention it in more detail a little bit later. This is called a hemiacetal. Probably a more apt description of that is that it's a half acetal, but nobody wants to call it a half acetal, so we use the word hemiacetal to describe that. And this then undergoes a further multi-step, in terms of the mechanism, conversion into the acetal. And that's an SN1 substitution reaction. And the byproduct of this process overall is a water molecule. So it's a condensation process. So we're going to go ahead and talk about in detail the mechanism for this transformation from carbonyl compounds like aldehydes and ketones into acetals. This is a reaction. You cannot do this transformation with esters or amides when there's a heteroatom attached to the functional group. But for aldehydes and ketones, especially for aldehydes, this is an important transformation. Let's go ahead and look in more detail at what's going on mechanistically in this important reaction. Okay, so the mechanism for acetal formation requires that we add an oxygen nucleophile to the carbonyl group. And so I told you that we need an acid catalyst to get good rates for this transformation. And the role of that acid catalyst is to protonate the carbonyl so that it can function as a good electrophile. Because hydroxy groups are not good nucleophiles. So we need this process of protonation of the carbonyl in order to generate a highly reactive form of the carbonyl in order to make this work. So let me go ahead and draw that protonated carbonyl. I'll just stick the proton right there. Now, when you see this O+, that's nothing like a carbocation. Don't ever attack an oxygen like that, an oxonium ion that has 3 bonds to oxygen. It already has an octet of electrons. If you count up the number of bonds, the 1 bond to the H and the 2 bonds to the carbonyl carbon, there's 8 electrons on that oxygen. Don't attack that oxygen. What we want to do is we want to attack the carbonyl carbon right here. Now, you use strong acids in these reactions because they don't permanently protonate the hydroxy group, but they make it easier to attack the carbonyl. So this is an essential step in order to allow this weak nucleophile, an alcohol, to attack. So you can attack that carbonyl carbon now that it's protonated carbonyl. And so This allows us to generate a mechanistic intermediate where we have that hydroxy group on there. We have the two methyl groups that were on the acetone, and we have this nucleophilic, the nucleophile, which is now an oxonium ion. It now carries a positive charge. And if you've been keeping track, we've been carrying along our conjugate base. That's the A-. I'll go ahead and draw it here. And that then picks up a proton from the nucleophile after it is added to the carbonyl group. And so now we end up with this neutral species. This is the hemiacetal intermediate that we've generated here. Let me go ahead and draw out that hemiacetal. It's got an alkoxy group and a hydroxy group. Again, that's the hemiacetal intermediate. Let me go ahead and label that for you. Hemiacetal. And there's still acid catalyst around, so let me go ahead and draw that acid catalyst. This time I'll draw it up here because I know that we need to catalyze the substitution of this hydroxy group. This is better, let me use my red pen. This is better than any SN1 type substitution that you might have seen earlier in this course, because the leaving group not only is a good leaving group, but we have this other oxygen lone pair to help push out that leaving group. And so let's take a look at that process. After we protonate this leaving group, we'll generate this tetrahedral intermediate that now has a great leaving group, and it has a lone pair down here, trying to push out that other oxygen atom. And now that it's protonated, you can't stop that H2O molecule or that component from acting as a leaving group. This is really the key step for this transformation, is getting this group to leave. So, and it's important for the mechanism of the reaction. So now we generate an intermediate that looks very much like this initial oxonium ion that we generated. Just like we said that this oxonium ion up here, this protonated carbonyl, is super easy to attack, in the same way it is super easy to attack, just as easy to attack this, this oxonium ion with 3 bonds to oxygen. And again, don't be tempted to ever attack that O+. That's not like a carbocation. It's got 8 electrons already, so there's no need to attack that. You want to attack this carbonyl carbon. That's how to get rid of that annoying positive charge that's bothering that oxygen atom so much. Oxygen's electronegative. It doesn't want positive charge, and the only way to get rid of it is to attack the carbonyl and give the electrons to the oxygen. So now we finally got these 4 bonds to oxygen and 2 heteroatoms attached. And if you look above and below, you'll see this similarity in these mechanistic intermediates here. I think you can tell what's going to happen. I'm going to take that conjugate base of our acid, and I'm going to deprotonate this oxonium ion here. And this is what leads us to our final product of this equilibrium process, our acetal. That's the mechanism for acetal formation. Let me go ahead and write acetal here since I took the time to write out hemiacetal. All of these reactions are reversible. They're completely reversible. You're just relying on equilibrium to drive this towards either acetal by removing water from the reaction, or if you wanted to, you could hydrolyze any acetal back, just add acid and add water. As long as you don't add acid, those acetals can be stable under amazing conditions. But in the presence of acid, this equilibrium is facile. So if I ask you the mechanism for acetal hydrolysis, it will involve exactly these same intermediates, the same 8 intermediates, and the only thing that will be different is arrow pushing. I'm not showing reversible steps for each of these arrows, but they're all reversible. And if I wanted to draw the mechanism backwards, I would just change these arrows to go in the reverse direction, and I would change all of my arrow pushing to match whatever's needed to go backwards. It's a good exercise. Take the time to try to think about this mechanism forward and back. Forward and backward, and it's important for this, an important mechanism for this chapter. Now, why would you want to take the time to make an acetal? And I told you already that if you want to synthesize acetals, really cyclic acetals are the ones that are going to give you the best yields and to be the most stable. Let me go ahead and show you a potential application for an acetal. Let's just suppose that you had this bromoaldehyde and you wanted to make a Grignard reagent out of that, so you could use it for CC bond formation. The problem that you're going to have is that as soon as you generate this Grignard reagent, it's going to attack the carbonyl within the same molecule. And even if you had even if it wasn't able to bend over like this and attack its own carbonyl, you always have more than one molecule in your flask. It would go find another substrate molecule like this one back over here and attack that instead. So how do we do Grignard chemistry or organolithium chemistry without messing up that aldehyde? And this is where acetal formation can protect the aldehyde. We can make an acetal intermediate as a protected aldehyde in the same way that we use silyl ether formation. Tert-butyl dimethyl silyl ethers are a way to protect alcohols so that they don't interfere with strongly basic conditions. So how would we make this into a stable acetal? Well, I already gave you a recipe. That would be to use our favorite diol for making acetals. This makes a stable 5-membered ring acetal. We need an acid catalyst for this. I'll write the word catalytic. You don't have to write the word catalytic, but I like to indicate that it's a catalyst for the reaction. And we're catalyzing acetal formation. This is going to make a 5-membered ring acetal. And here's our 5-membered ring acetal. And there's our ethylene glycol. That's the name for that diol molecule. And that's the structure of the cyclic acetal we generate. Now we can do all kinds of strongly basic chemistry on this side of the molecule, and none of that is going to touch that acetal. It won't be bothered at all by strong bases or strong nucleophiles. No strong base that you know and no strong nucleophile you know will mess up or react with that acetal. Acids, though, that's the way to take those off. So let's go ahead and try to do some nucleophile chemistry. If we converted this carbon-bromine bond into a strong nucleophile by adding magnesium, that's the way we'll generate a Grignard reagent. I think Sapling often writes ether down below as a reagent for Grignard formation. I don't require that simply because our current textbook doesn't mention that. So let's go ahead and what happens when we add magnesium to an organobromine compound. You convert that into a super nucleophilic carbon-magnesium bond. And we still have our acetal on there. No problem having an organomagnesium reagent in the same reaction as an acetal because acetals are stable. Now, as soon as I generate a Grignard reagent, I want to use that as a nucleophile, don't you? And make it attack a carbonyl. I'm going to pick a very simple carbonyl here. Let me go ahead and pick acetaldehyde as an example. That will be step 1. You know what step 2 is. I'm going to follow this up with an aqueous workup. Step 2, I'll just write H2O, but that's the workup for the reaction. And so what would happen here is we would end up generating an alcohol out of this reaction. So let me go ahead and draw out that, that same framework before I get ready to, there we go. And I'm going to take this carbon atom right here, and I'm going to draw out that new bond that I generate. That's the 2-carbon piece right there that used to be part of our acetaldehyde. And we protonate that alkoxide with our aqueous workup. Now I'm ready to regenerate my aldehyde. And as long as I've kept this system free of acid, I can come along with some sort of a strong acid like HCl or catalytic toluene sulfonic acid or catalytic sulfuric acid, and then hydrolyze that carbonyl. So I'll write hydrochloric acid, but you could also use catalytic toluene sulfonic acid just like you used for acetal formation. I'm picking HCl because it's a cheap acid and you're doing it in water. And we already have bottles of HCl in water sitting in the lab. You don't even have to bother making a new solution. And that hydrolyzes back the acetal. I got to make sure I get the number of carbons right here, 2, 3, 4. And then here's my aldehyde. There it is. I get it back. Now you also get back the diol or the alcohol that you started with. We usually throw that away. We're not trying to keep ethylene glycol. We're trying to take this aldehyde and do something else with it. So that's a pretty powerful transformation. I don't know if you're like me, but I feel like I want to protect that OH with a silyl protecting group and then do more Grignard chemistry or organolithium chemistry adding to the other side. That's the way a synthetic organic chemist thinks, is protect things and then work on another part of the molecule as long as you've got either an aldehyde protected as an acetal or a hydroxy group protected as a silyl ether. Very common sequence of transformations to protect and then do nucleophilic chemistry. Okay, whenever you make cyclic acetals, they are way more stable and you get much higher yields than if you're just making acyclic acetals with methanol. And I want to point out that if you have compounds that have hydroxy groups and aldehydes in the same molecule, they can generate hemiacetals that are stable. Cyclic hemiacetals are stable if they're part of a 5 or a 6-membered ring. And let me go into a little more detail on that issue. So if you take a hydroxyaldehyde, it's going to be slamming into that carbonyl, colliding over and over and bouncing off. Every once in a while, it'll form a bond. And those collisions are much more frequent when the hydroxy group is physically attached to the aldehyde. And so that's why you get at equilibrium a higher ratio of this hemiacetal. That's why the equilibrium constant tends to favor the cyclic form, the hemiacetal form, when it can form a 5 or 6-member ring. It's because collisions are more frequent. Somebody has gone through the trouble of measuring the ratios of these two species at equilibrium. And you always reach equilibrium very quickly, in less than a second, whenever these molecules are in any type of a solvent in solution. So I'm going to go ahead and make a table here where we take a look at these equilibrium amounts as we vary the potential ring size. So I'm going to call these two structures either closed, maybe I should call it, whoops, I can't even spell closed, closed or open. But maybe I should call it hemiacetal. I'm using closed because it's probably easier for you to understand than if I say acetal at this stage. But that's the hemiacetal form. When I say percent closed, I'm saying how much of this is in the hemiacetal form. Well, it depends on the ring size. If I have a hydroxy group over here and I'm trying to make a 4-membered ring, that's 90-degree bond angles in the 4-membered ring, and carbon does not like 90-degree bond angles. It likes 109 degrees. So if I try to make a 4-membered ring by cyclizing, less than 1% of my hydroxyaldehyde will exist in this cyclic form. It doesn't want to be a strained 4-membered ring. But if I have the hydroxy group over here where this carbon is, that very much likes to cyclize. A 5-membered ring is pretty good and kinetically very favored. 89% of my hydroxyaldehyde will exist in this cyclic 5-membered ring form if the hydroxy group is poised 5 atoms away from the carbonyl. And if, as I've shown here, it can cyclize to make a 6-membered ring, this particular molecule right here can cyclize to make a 6-membered ring, that's almost all exists as the hemiacetal. It's in equilibrium, it's in rapid equilibrium, and at equilibrium, it's 94:6 ratio. Out of every 100 molecules, 94 of them will be in this closed form. So, so you get a sense that for cyclic hemiacetals, those can be quite stable. Anything bigger than a 6-membered ring is not very good. You know, it depends on the ring size, but it'll be less than 20% in the cyclic form. So it's really only these 5 and 6-membered rings where you should expect hemiacetals to be stable and in their cyclic form. Let's go ahead and take a look at the mechanism for hemiacetal formation. There's no surprises here. It's going to be just like that acyclic version, but let's go ahead and walk through that. I'm going to go ahead and draw out this substrate here. And when I draw out the acid catalyst, since I'm drawing an arrow pushing mechanism, I'm going to symbolize that acid by using this symbolic HA. And of course, that's going to protonate the carbonyl under these conditions. There we go. You protonate the carbonyl. Because if you don't protonate the carbonyl, it's hard to get this OH to attack. Now, I think the book discusses base catalyzed acetal formation and base catalyzed addition of water to, that's not important for any of the reactions that we're going to talk about this quarter. When you put bases in the presence of aldehydes or ketones, the dominant chemistry is deprotonation next to the carbonyl to form carbanions. Nobody cares about acetals or hemiacetals under basic conditions. It's unclear to me why the book discusses that. I wouldn't ask you that, but acid catalyzed acetal and hemiacetal formation is important. So let's go ahead and draw out this protonated carbonyl, because when you've got protonated carbonyls, they're extremely reactive. So here's that protonated carbonyl. I really want to attack that. And even a lame nucleophile like a water molecule or a hydroxy group can easily attack a protonated carbonyl group, especially when that oxygen atom is 6 atoms away. And so this generates a ring. Here we go. And we still have that proton on there. And this is where, if you'll recall from this first step, we generated A- as a conjugate base. We didn't really use that here, but in this step, that's going to come in very handy because we can deprotonate that oxonium ion. We can deprotonate this protonated oxygen, pull the proton off, and that's what generates our hemiacetal. And this hemiacetal, as I've shown you above, is pretty stable. All of these reactions are reversible. When I tell you that this very quickly, in less than a second, will generate this equilibrium 94:6 ratio, all of these steps are happening in that time period. Even though the mechanism is long, it's an extremely fast process, as long as you have an acid catalyst around. Particularly important for that. Okay, acid catalyzed, hemiacetal formation, acetal formation. You need to protonate that carbonyl in order to get oxygen nucleophiles to add to carbonyl groups, because hydroxy groups are just not good nucleophiles. So you really need to help them out. Now I'm going to show you a particular SN1 transformation. Yes, we're kind of going back to SN1 reactions, but this is a special one. So whenever you take these cyclic hemiacetals, like with the 6-membered ring or the 5-membered ring, and you expose them, I'll give this one a stereogenic center. It doesn't even matter whether it's racemic or not. And you expose these to catalytic acid and alcohols. You've seen this done with T-butanol or things that can form stable carbocations. Earlier in the year in this course, I'm going to use methanol as a nucleophile, and it's being used as a solvent. I've indicated that under the arrow. Now, the HCl is going to protonate, well, it'll protonate every oxygen in here. Protonate, deprotonate, protonate, deprotonate, protonate, deprotonate. But every once in a while, when you protonate the water, it acts as a leaving group, and it will get replaced by the methoxy. And when the methoxy group leaves this system, it's going to act as a, it's going to be attacked from either face when you generate this carbocation type intermediate here. There's two different faces. Half of the time, the methanol is going to add from the top face. I'll go ahead and draw it like this. The other half of the time, I'm not going to write plus E just because I want you to see explicitly. The other half of the time, the methoxy is going to add from the bottom face, and you have no control over that. That's a particular characteristic of the SN1 reaction, this idea that you can attack from either face of a carbocation or a carbocation-like intermediate. That's nothing new. So you could have written plus E here, as long as you write one of those explicit enantiomers. But it doesn't matter which isomer we start from, we're going to get a mixture of the two. Whether we start with a mixture or start with hydroxy up or start with hydroxy down, doesn't matter, because you can't control whether methoxy adds from the top or the bottom face. Let's take another example. This time I'll draw a 5-membered ring. Cyclic acetal, where I've already got an ethoxy group on here. And if I don't indicate a stereochemistry, it implies that I've got a racemic mixture there. But I usually, when I ask you to draw the product for me, I ask you to clearly indicate stereochemistry. But oftentimes synthetic organic chemists don't just leave it as a line, because within the community, we recognize stereochemistry. We've already taken our courses in organic chemistry. So when you expose this to catalytic HCl, let me write catalytic up here. We're not trying to substitute anything with chloride. We just want to use it to help assist the leaving group. And so I'll write catalytic HCl again here. Again, I'm not trying to substitute anything with chloride. What we really want to do is substitute that ethoxy with a hydroxy group. So instead, I'm going to add water as my nucleophile to trap out the cationic intermediate that we generate in this SN1 reaction. And so in this particular case, no matter what enantiomer I got, I'm going to get a mixture of 2 enantiomers here for my product. One of them where the hydroxy group is up, and the other one where the hydroxy group is down, right? You'll get a 50-50 mixture in there. Now in this particular case, we're also going to have an ethanol leaving group. Let me go ahead and indicate that there, plus ethanol. So we get the two, you get the 2 enantiomers, plus you get the ethanol leaving group. Now why would we talk about this type of chemistry? It turns out that the molecules with these kinds of structures called glycans, so monosaccharides, disaccharides, are particularly apt to form these kinds of structures. And I even showed you these types of organic sweeteners that are present, mogricin, that are present in the artificial sweeteners like stevia or monk fruit sweetener. These types of bonds are particularly important for joining sugars to other types of molecules like steroids. Those are called glycosides whenever you make these kinds of bonds. Now, I've indicated down here that one of the reasons why they teach this stuff in chapter 21 is there's another chapter in the book that is completely devoted to these classes of molecules, monosaccharides, sugars, that have these acetal type linkages, these mixed acetal linkages. We're not going to cover that chapter this quarter, and in the past we have. So it used to be particularly important to cover this, and I'll leave it up to you to just go glance at that chapter just to convince yourself that this is an important type of chemistry, this formation of acetals and mixed cyclic acetals. Let's go ahead and take a look at the mechanism for that process of forming these mixed cyclic acetals through SN1 reactions. Why is it so interesting this time, and it wasn't an interesting reaction 2 quarters ago when you were taking Chemistry 51A here at UC Irvine? Let me go ahead and draw the mechanism for this reaction. Remember, the reaction involves a catalytic acid. If I'm drawing an arrow pushing mechanism, I'm going to symbolize that using HA, because I don't really know whether that's H3O plus or HCl or some protonated species. I don't even care. So the key step en route to the substitution product, let me switch to my red pen here, is protonation of this external hydroxy group here. It's also protonating over here on this oxygen atom. You protonate, you deprotonate, you protonate, you deprotonate, you protonate this one, you deprotonate, over and over again. But when you I'm only going to show the protonation of this hydroxy group, even though protons are flying all over the place, because that's the one that leads to the product. I'm only showing the steps mechanistically that lead to the product, the substitution product. So when we do that, we end up generating a great leaving group here, H2O plus. Please don't attack that oxygen. It's got 8 electrons. So if you want to attack something, and this is the key step here, if you want to get rid of that positive charge, let's get this to act as a leaving group. But this isn't just your regular SN1 ionization reaction. Not only is this leaving, it's being pushed out. Let me go ahead and draw the fact that there's lone pairs on this oxygen atom. And those lone pairs are pushing. They're pushing this leaving group out. You're never going to find a closer nucleophile than that. So this isn't just a regular SN1. This is a great ionization reaction. Fantastic. Better than forming any T-butyl carbocation or any type of SN1 reaction that you might have seen before. This is a special one. Why is this such a good cation, whereas T-butyl carbocations are not? Because every atom in this molecule has a filled octet of electrons. That oxygen may have a positive charge, not so great, but it's got a filled octet. It's got 8 electrons. That's why that's a better SN1 ionization than anything from a previous quarter you might have seen. Let's go ahead and draw the formation of that new oxygen-carbon bond. I'll get my alcohol to come in and attack. And there we go. Boy, it just feels so good to attack these kinds of, it looks like a carbonyl that's got an O+. Even an oxygen nucleophile can attack those types of carbonyls if there's a third bond on that oxygen and a positive charge. So the next intermediate that we generate now has this oxonium ion. And if you've been keeping track, there was this conjugate base that was generated, right? We've got to have a counter ion here. It started off neutral. Everything has to still add up to 0 overall net charge here. Now we can use that conjugate base from our acid to pull that proton back off. That's why the acid is catalytic. We regenerate the acid in the last step of this arrow pushing mechanism. And so now we generate this mixed cyclic acetal. Boy, that looks like a sugar. If you decorate this with hydroxy group, that's a sugar molecule, like glucose, like derivatives of glucose. So really exciting chemistry because it relates to the entire field of glycans, which are important for extracellular proteins. About half of the extracellular proteins that exist in your body are floating around with sugars attached to them through this type of chemistry. Okay, again, these types of mechanisms will be important for a later chapter. That's all we have to cover here for chapter 21. I cannot wait to get to chapter 22 because we're going to talk about all kinds of derivatives of carboxylic acids. Acyl chlorides, remember those things that we use to do electrophilic aromatic substitution? Those are going to be money reagents for this coming chapter. We'll talk about amides, esters, hydrolysis of those. Great stuff coming up. We have an exam coming up on Monday. It's going to be in the online sapling system. So go back and practice every reaction you can get your hands on. My exam will consist of reactions you've never seen before, chemistry you've never seen before, questions you've never seen before. And the only way to get ready for that is to hone your tools by working lots of problems. All right, I'll see you on Monday.